Where is the memory of your mother's face right now? Not the image, the actual physical trace of it. In 2016, the Salk Institute calculated that your brain holds at least 1 petabyte of data, roughly the storage capacity of the entire internet circa 2000. Yet no surgeon has ever cut open a skull, pointed to a cell and said, "There, that's where Tuesday's lunch is stored." Your memories are inside you. But the question of where, exactly, is one that modern neuroscience still cannot fully answer. Most people assume memories work like files on a computer. You experience something, your brain saves it somewhere, and when you need it, you pull it back up. Folder by folder, neat, organized. That model is wrong. It has been wrong for decades, and the more we learn, the more wrong it becomes. For most of the 20th century, scientists believed memories lived in synapses, those microscopic gaps between neurons where chemical signals pass back and forth. Strengthen a synapse, store a memory. Weaken it, lose the memory. Simple, elegant, and incomplete. Here is what shook that idea. In 2014, David Glanzman and his team at UCLA took marine snails called Aplysia and gave them a series of mild electric tail shocks. The snails learned to retract their bodies defensively, a basic form of memory. Then Glanzman's team erased the synaptic connections that supposedly held that memory. They were gone, dissolved. But when the neurons were stimulated again, the memory came back, fully formed, as if it had never left. If the memory was stored only in those synapses, that should have been impossible. Glanzman's conclusion was blunt. Memories are not stored where we thought they were stored. The filing cabinet model is a comfortable fiction. So if your memories aren't neatly shelved between two neurons like a book in a library, where are they? The answer involves places inside your body that, until very recently, no one thought to look. And what researchers are finding there is challenging the most basic assumptions about what a memory actually is. To understand where your memories go, you first have to understand what a memory physically is. Not the feeling of remembering, the actual thing, the biological object. Scientists call it an engram. The term was coined in 1904 by the German biologist Richard Semon, but it took over 100 years before anyone could actually see one. In 2012, Susumu Tonegawa's lab at MIT's Picower Institute did something extraordinary. Using a technique called optogenetics, where specific neurons are genetically modified to respond to light, they identified a small cluster of cells in a mouse's hippocampus that activated during a specific experience. When they later reactivated those exact cells with a pulse of light, the mouse behaved as though it was reliving the original event. They had found the engram, or at least part of it. Think of it this way. Imagine your brain as a city at night. An engram is not one building with the lights on. It is a specific pattern of buildings, scattered across different neighborhoods, all flickering in a coordinated sequence. One cluster fires in your hippocampus, another in your visual cortex, another in your amygdala. The memory is not in any single location. It is the pattern itself, distributed across your brain like a constellation of stars that only makes a shape when you see them all at once. In 2020, Tonegawa and Sheena Jocelyn of the University of Toronto published a comprehensive review in Science describing what they called Engram complexes. Their research showed that a single memory recruits cells across multiple brain regions simultaneously. These cells are physically altered by the experience. They grow new synaptic spines, change their excitability, and form new circuit connections with other engram cells. Your brain literally reshapes itself around every experience you have. But here is what gets strange. Tonegawa's team also showed that engrams can exist in a silent state, physically present in neurons but impossible to recall naturally. In mouse models of early Alzheimer's disease, they found that memories were still there, encoded in the cells. The mice just couldn't access them. The filing cabinet wasn't empty. The drawer was jammed. The research is still evolving on this, but the implication is staggering. Some of your memories that you believe are lost, childhood events, names, faces you think you've forgotten, may still exist physically inside your neurons. The cells that hold them may still be alive, still connected, still carrying the trace. You haven't lost the memory, you've lost the key to the room it's sitting in. And yet, even this picture is not complete. Because Tonegawa's engrams are made of neurons, and there are cells in your brain that outnumber neurons and do something with memory that nobody expected. That discovery happened just weeks ago. In May 2025, a team at MIT, led by Dmitry Krotov and Jean-Jacques Slotin, published a paper in the Proceedings of the National Academy of Sciences that may have rewritten the rules. The paper focused on astrocytes, star-shaped brain cells that were dismissed for most of the 20th century as mere support staff. Janitors, custodians, cells that cleaned up around neurons and kept blood flowing. Nobody thought they computed anything. That assumption now looks like one of the biggest oversights in the history of brain science. Your brain contains roughly 86 billion neurons. It also contains billions of astrocytes. Each one extends thousands of thin tentacles, called processes, that wrap around individual synapses. A single astrocyte can touch up to 100,000 synapses at once. That means one astrocyte is listening in on the conversations of hundreds of thousands of neuron connections simultaneously. If you've ever had a moment where a smell, coffee, rain, a specific perfume, instantly pulled back a memory you hadn't thought about in 30 years, and you wondered how your brain could retrieve something that old and that specific that fast, this may be why. The MIT team built a mathematical model showing that astrocytes don't just listen, they compute. Using calcium signaling, a slow, chemical form of communication entirely separate from electrical nerve impulses, astrocytes coordinate information across massive numbers of synapses simultaneously. The model demonstrated that a neuron-astrocyte network can store dramatically more memories than a network of neurons alone. Not slightly more, orders of magnitude more. Enough, in fact, to account for the brain's estimated petabyte-scale storage capacity, something that neuron-only models could never explain. Leo Kozachkov, the paper's lead author, described it this way. By careful coordination of the spatial temporal pattern of calcium in the cell, and then the signaling back to the neurons, you can get exactly the dynamics you need for this massively increased memory capacity. Think of neurons as the musicians in an orchestra. Now think of astrocytes as the conductor. The musicians produce the sound, but the conductor decides which instruments play when, how loudly, and in what combination. Without the conductor, you get noise. With the conductor, you get a symphony. Your memories may be that symphony, not stored in any single neuron, but orchestrated across vast networks by cells that nobody thought were doing anything important. If this is making sense, and you want the part about what this means for your memories as you age, subscribe now, because what comes next is the section almost nobody talks about, and it arrives in 4 minutes. Some scientists dispute this model. It is, after all, based on computational simulation, not direct observation in living brains. But the architecture fits. And if it's right, it means that memory isn't just an electrical phenomenon. It is also a chemical one, a structural one, something woven into the very geometry of cells that we ignored for 100 years. But none of this answers the most disturbing question. What happens to a memory when the neurons that hold it die? What almost nobody talks about is this. Your memories might not be limited to your brain cells at all. In 2018, David Glanzman at UCLA conducted an experiment that should have made front-page news everywhere. He extracted RNA, ribonucleic acid, the molecule that carries genetic instructions inside cells, from the nervous systems of marine snails that had been trained to respond to a shock. Then he injected that RNA into untrained snails. The untrained snails immediately behaved as if they remembered the training. They contracted defensively for an average of 40 seconds, nearly identical to the trained snails. The control group, injected with RNA from untrained snails, contracted for about 1 second. Read that again. A molecule was extracted from one animal's nervous system, placed inside a different animal, and that second animal behaved as though it had the first animal's experience. Glanzman was direct. It's as though we transferred the memory. Glanzman believes memories are stored not just in synapses, but inside the nucleus of neurons, in the epigenetic marks on DNA. These are chemical modifications, primarily a process called DNA methylation, where small molecular tags attach to your genetic code and change which genes are turned on or off. Your DNA is not just your blueprint, it is also, possibly, your diary. Every experience you have, a broken bone, a sleepless year, the death of someone you loved, may leave a molecular stamp on the DNA inside your neurons. Depending on who you ask, this idea is either the future of memory science or a controversial hypothesis that needs far more evidence. But the data points keep accumulating. At the University of Utah, researchers have demonstrated that epigenetic modifications, specifically DNA methylation and histone modification, stabilize gene expression patterns that are critical for long-term memory storage. The modifications persist long after the original experience. They are, in a very real sense, the body's way of writing experience into chemistry. Now take that idea one step further. In 2000, Gary Schwartz and Linda Rusick at the University of Arizona published a study documenting personality changes in heart transplant recipients that paralleled traits of their donors. One recipient, a 47-year-old white man who received the heart of a 17-year-old black teenager, suddenly developed an obsessive love of classical music. The donor had been a violinist. Historians still argue whether these cases represent genuine cellular memory or psychological confabulation. The research is controversial, but across a sample of 10 cases, Schwartz found parallels in 2 to 5 personality changes per recipient that matched the donor's profile. If even a fraction of this is real, if RNA carries behavioral traces, if DNA records experience, if cells outside your brain retain some shadow of who you are, then the question, where do your memories go, doesn't have a single answer. They may go everywhere. Into the chemistry of your cells, into the structure of molecules, into regions of your body that no one thought had anything to do with remembering. So the question becomes, if your memories are this distributed, this embedded in your biology at every level, what happens to them when you sleep? Here is what the research actually tells us about protecting and strengthening the places where your memories live. Not theory, not speculation. Specific actions tied to specific mechanisms. Step 1. Sleep between 7 and 8 hours per night and prioritize the hours before 2 a.m. During slow-wave sleep, the deep, dreamless phase that dominates your first 4 hours, your hippocampus replays the day's experiences at high speed. Researchers at MIT's Picower Institute have shown that during this phase, the hippocampus sends coordinated signals to the cortex in a process called systems consolidation. Your short-term memories are literally transferred to long-term storage. If you cut your sleep to 5 hours, you gut this process. Your brain has experiences to file, but no time to file them. Step 2. Move your body for at least 30 minutes a day at moderate intensity. Walking counts. Physical exercise increases a protein called BDNF, brain-derived neurotrophic factor, which directly promotes the growth of new synaptic spines, the structural basis of Engram formation. A 2011 study led by Kirk Erickson at the University of Pittsburgh showed that 1 year of moderate aerobic exercise increased hippocampal volume by 2% in adults with a mean age of 67. That reversed age-related volume loss by approximately 1 to 2 years. Step 3. Learn something genuinely new at least twice a week. Not a crossword, not Sudoku. Something that forces your brain to form new Engram complexes. A language, an instrument, a manual skill. Tonegawa's research has shown that Engram cells are recruited from pools of neurons with higher excitability. The more novel the stimulus, the more excitable the neurons become. Repetition of familiar tasks does not produce the same effect. Step 4. Eat foods that support astrocyte function. Your astrocytes depend on omega-3 fatty acids for their membrane integrity and on magnesium for calcium signaling, the very mechanism the MIT team identified as central to memory orchestration. Fatty fish, walnuts, dark leafy greens, and pumpkin seeds are among the most direct dietary sources. Dr. Lisa Mosconi at Weill Cornell Medicine has been prescribing omega-3 rich diets to patients over 60 since 2018, specifically citing their role in supporting glial cell health. Step 5. Reduce chronic stress. Cortisol, the hormone your body releases under sustained stress, literally shrinks the hippocampus over time. A 2018 study in neurology by Justin Echufo-Chugi at Harvard Medical School tracked 2,231 adults with a mean age of 49 and found that those with the highest cortisol levels had measurably smaller brain volumes and performed worse on memory tests. Stress is not an abstract threat to your brain, it is a physical one. If even one of these steps was new to you, hit like. It tells the algorithm to show this to more people who need it. Step 6. Practice deliberate recall. Don't just consume information, force yourself to retrieve it. At the end of every day, sit quietly for 2 minutes and try to reconstruct in detail 3 things that happened. What you ate, who you spoke to, what the weather was. This practice, which cognitive scientists call retrieval practice, strengthens the pathways between engram cells and makes silent memories more accessible. Here is what physics and biology, taken together, are quietly telling us. Your memories are not files. They are not stored in one place. They are patterns, constellations of cells, molecules, and chemical marks that stretch across your brain and possibly beyond it. They live in the electrical firing of neurons, in the calcium waves of astrocytes, in the methylation tags on your DNA. They are consolidated during sleep, strengthened by movement, and eroded by stress. They can fall silent without disappearing. They can be transferred in molecules between organisms. They can persist in cells long after the connections that seem to hold them are destroyed. The old model, experience goes in, memory comes out, stored neatly in a synapse, was never true. The real picture is stranger, more distributed, and more beautiful. Your body is not just carrying your memories. In some deep molecular sense, your body is your memories. Every cell holds some fragment of what you have lived through.
Science doesn't know the full answer yet. It may not know it for decades.
But what the last 10 years of research have made clear is that the question, where do memories go, cannot be answered by pointing to a single structure or a single mechanism. They go everywhere, into everything you are. I want to hear from you. Drop a comment below. What is one memory you would give anything to have back in perfect detail? And what would it mean to learn it might still be there, somewhere inside you, waiting? Your body is not a storage device. It is the memory itself. If someone in your life is struggling with memory loss, or just wondering what happens inside them as they age, share this with them. They deserve to know what science is actually finding. And if you haven't subscribed yet, you should. What's coming next week is a question I've been turning over for months. What does your brain do in the 6 minutes after your heart stops? The data on that is extraordinary. Your memories don't leave, they change form. And so, quietly, do you.